But how do the different types of cell division relate to the variability of traits in humans? So in mitosis, this it's really complicated because you are creating two clones, right? You're forming two identical data cells from one cell. So this process is so critical because we're just cloning. So when mitosis is not regulated correctly, health problems such as cancer can result. So this mitosis is really hard to have this variability of traits in humans. So let me put here really hard. It's not really hard. I find it even impossible because the the only thing that can happen is for you getting cancer or Down syndrome, as we saw in the procedures, in the stages. So it's not a good thing if we have variability mitosis. But in meiosis, let me change the color so we can understand. Meiosis, it's a completely different thing. So the gametes in the telophase. Remember that we had this phase that happened in the telophase. They was separating the cells that I didn't know how to pronounce in the videos. So these cells form gametes. They're haploid, but they're not genetically identical. This will make us have variability. So why do we get many genetically different gametes? So there are two main reasons: the crossing over. You remember when I think it was on which I think it was in the pros phase of meiosis. I think it was in pro phase. We had this crossing over. So this would be something like this. So we have the the chromosomes. It would do something like this. So this the crossing over. So these points exchange genetic material and they're chose more or less random. So we get different variabilities because of this. And in meiosis happens many times. And it can happen in many different points. So for me, as I said, in one of the stages of meiosis, for me, it's I'm counting that there are infinite codes or different variations that we can find, that we can have. So it's pretty excited. And then we have the random orientation of homologous pairs. I don't know if I talked to in the stages, but as I think I talked to that the pairs when we're separating in the first meiosis part, it's not they're not chosen randomly. I said that these are chosen randomly, so this is you could have more genes of your mother than your father. You could have crossing overs and stuff. I told that. So this also happens, and this affects more variations of the traits in humans. So these random orientations just increase our probability of having more variables. And that's a fun fact. In a human cell, the random orientation of homologous pairs alone allows for over 888 million different types of possible gametes. So, for me, that's insane. There's so many different possibilities. and we can have so many different types of possible gametes just by this random orientation. So, and even if we combine these both, we are like the number of genetically different gametes that you can make, or like you, like the person reading this, or the person listening to this, probably Mr. Miller, can make it effectively infinite. So, like, if we combine those, infinite, we have almost infinite amounts of variation. So, like, this is why it's like so messed up. For me, so messed up in the head that, like, I could have, for example, blue eyes if, like, in the stage of prophase, except of having what? This? Except of having this, I could have this, and this made a blue eyes person. So like it's pretty crazy to think that, oh, maybe I could have blonde hair, like I could have um when I was created I could have blonde hair if like this chromosome just like crossed over with the other chromosome, and this gamlet or whatever it's pronounced made a blonde type of hair. So like it's pretty like still doesn't fit in my mind, but like it's pretty awesome to think about it.